All right, let me pull up the slides here and you guys can tell me if I remember correctly. I think we at least got through mitosis and meiosis. Does that sound right? Did we talk about meiosis in males and females?
Yes.
Okay. Did we talk about chromosomal abnormalities?
Yes.
Did we talk about non-disjunctions?
No.
Okay.
Uh,
All right.
wait. Yes, yes, I think we just finished here though.
Okay, perfect. Well, let's go ahead and review this slide just for good measure. So we are still on chapter 3, ladies and gentlemen. This is the end of chapter 3. We only have probably about 10, maybe 15 minutes left to talk about chapter 3, and then we'll delve right into 4. Just don't forget, guys, these recordings are available to you. If you miss the details or if you prefer to sit back and just listen, these recordings are available to you if you need them. Also, recordings from previous semesters are available to you in the module. So, for some reason you want to sit back and just enjoy or be able to ask questions or a combination of both, whatever you would like is totally fine. So non-disjunctions are basically errors that happen when the chromosomes fail to separate properly during cell division. So thinking back to what we learned about mitosis, when we learned about mitosis, we learned about anaphase. So remember, interphase was the pregame where all the DNA was replicating, the organelles are replicating. Prophase was in that DNA was coiling around proteins called histones to form visible chromosomes. And then metaphase middle, that's when all the sister chromatids are aligned along the equator of the cell, nice and neatly by the spindle apparatus. And then anaphase, that's when the spindle apparatus is winning its tug of war and successfully pulling apart those sister chromatids. Okay. So when that's what should happen, disjunction is what we want. We want disjunction. So non-disjunction is an error that occurs when it fails, when those spindle apparatus, generally just in one location, fail to properly separate those chromosomes during cell division. So when a non-disjunction occurs during meiosis, remember meiosis is the formation of gametes. If this non-disjunction occurs during meiosis, then the resulting gametes will have an improper number of chromosomes. So you might end up with a sperm cell or an egg cell that has an extra or a missing chromosome, for example. Let's just say that sperm cell has an extra or has two 21s, and then the egg cell only has one. But once that egg and sperm combine, you now end up with 3 of the number 21 chromosomes. So disomy is what we want. Disomy is the typical or quote unquote normal condition, at least when we're talking about humans. And this is a class on humans, so we'll focus on humans. So disomy refers to the 2 chromosomes that make up each pair. So we have 22 pairs of autosomes, 1 pair of sex chromosomes. So break that word up. Di means 2, somy means chromosome. So disomy is the standard condition. Mono, break up that word. Mono means 1, somy means chromosome. So a monosomy is where there's a non-disjunction where one of the chromosomes or one of the sex cells, is missing a chromosome. So either the sperm or the egg cell is missing a chromosome. So an example of this would be XO syndrome. So that's a female that has 1 X chromosome but is missing the second X. That's called Turner syndrome. And then a trisomy is the other kind. So a trisomy, break it up. Tri means 3, somy means chromosome. So a trisomy occurs when an extra chromosome is present. So for example, maybe the egg cell had a non-disjunction and there's still 2 21 chromosomes. So when it combines with the sperm cell, you end up with 3 number 21 chromosomes. So that's trisomy 21, or more commonly known as Down syndrome. So even though Down syndrome is a debilitating, severe, life-altering genetic disorder, it is survivable, of course, but just so you know, that is one of the most survivable of them. Trisomy 11, I believe, is Edwards, and trisomy 18 is Pateau. I always switch the two, so don't quote me on that. I'd have to look it up to remember. I should put this on my slides so I don't forget all the time. But they're both survivable, but very severe genetic disorders. If you're taking the lab, we do go into more detail on these non-disjunctions, and the lecture we just kind of introduce the topic. But if that's something that interests you, definitely take the lab class. All right, so questions about non-disjunctions before we move on. Feeling okay? Okay, so let's talk a little bit about genes. So we already dove into this topic quite a bit on Monday when we did Punnett squares. We looked at, essentially, the various genotypes. We looked at dominant and recessive alleles, and we looked at various genotypes, and we used Punnett squares to calculate the likelihood of various combinations of genotypes and phenotypes, and it, depended upon the scenario. But we looked at the monohybrid crosses, we looked at ABO blood typing, and we also looked at sex-linked traits. So there's, a lot of genetics is far more complex than that, and we'll talk a little bit about that here in a moment. But just like we have types of chromosomes, just like we have types of cells and we have types of cell division, there also will be types of genes. So some genes are structural genes, which are responsible for forming our bodily structures, just like it sounds. And then some genes are regulatory. So regulatory genes are responsible for turning other genes on and off. So those are sometimes called Hox genes. When you're reading about genetics, you might hear the term Hox gene. So for example, just because you might have the gene that makes you more likely to later develop breast cancer doesn't necessarily mean you're going to develop breast cancer. There are genes that get turned on and off depending upon environmental circumstances. The field of epigenetics is really looking into understanding how the environment influences whether these genes are swapped on or off. So exciting stuff to come there. I don't necessarily have all the answers for you guys there, but just so you know, that's what they mean when they say a Hox gene. It's something that turns other genes on and or off. All right, polymorphism. So a little bit of vocab. This may hopefully sound a little bit familiar from what we did on Monday. But the image there at the bottom of the slide, those are chromosomes. Okay, so you're looking at a homologous pair of chromosomes there. So that band, that purple and white band, that's representing the allele that codes for either purple or white flowers. Okay, so at that allele, at that location on the chromosome, essentially that's the location that determines whether that flower is purple or white. Okay. So a locus is the location, the physical location of that gene on the chromosome. That will be important. That will continue to come up. So a locus is the location of the gene on the chromosome. Alleles are just different variations, or you can think of them as flavors. So flavors of a gene. So in this case, let's say, for example, that purple, the allele that codes for the purple color is dominant over the allele that codes for the white color. If that plant inherited a purple allele from one parent and a white allele from the other, the overall color of that plant would be purple. Okay. If we're talking simple monohybrid. Now, sometimes there's blended inheritance, like sometimes both the purple and the white allele may be expressed in the phenotype. So if you get a purple allele from one parent and a white allele from the other, that plant may be purple with some white speckles. But just know that the purpose of this slide is just understanding that the locus is the physical location of the allele or the gene. All right, this slide is also a little bit of review from Monday, a little bit of vocab. So genotype, so think genogenetic, phenophysical, genogenetic, phenophysical. The genotype is the letters. The genotype is the genetic makeup of that organism. So when we're talking simple monohybrid crosses, there's only 3 genotypes we'll talk about: homozygous dominant, heterozygous, or homozygous recessive. So regardless of what trait we're talking about, whether it's earlobes or tongue rolling or PTC tasting or widow's peak or hitchhiker's thumb, no matter what you're talking about, 2 uppercase letters will always represent homozygous dominant. Okay, you break that word up. Homo means same. Zygous means zygote. So remember, a zygote is just a fertilized egg. So it's referring to the 23 chromosomes that you get from mom's egg and the 23 chromosomes you get from dad's sperm. So all of us are quite literally 23 chromosomes from mom, 23 chromosomes from dad, and the combination. So at that locus, if we inherit a dominant allele from both mom and dad, then we are homozygous dominant for that trait. So always represented with 2 uppercase letters. At that locus, if we inherit 2 recessive alleles, if we inherit a recessive allele from both mom and dad, then we are homozygous recessive, and we have 2 copies of that lowercase recessive allele. If we are heterozygous, so break up that word, hetero means different, zygous still means zygote. So that just means that you inherited a dominant allele from one parent and a recessive allele from the other. So heterozygous will always be represented with one capital letter and one lowercase letter, okay? So homozygous, there's two types: homozygous dominant, two uppercase letters; homozygous recessive, two lowercase letters. Heterozygous is always represented as one uppercase and one lowercase, okay? So if I'm asking for a genotype, that's what I'm asking for. I'm asking for homozygous dominant, homozygous recessive, or heterozygous. If I'm asking for a phenotype, I'm simply asking for the physical expression. So it depends on what we're asking about, right? Are we asking about earlobes? Are the earlobes attached or free hanging? Are we asking about tongue rolling? Can that person tongue roll, or are they unable to tongue roll? Can that person taste PTC? Do they have a hitchhiker's thumb? Do they have a widow's peak? Things like that. So that's what I'm asking when I say phenotype. What is the physical manifestation of that genotype? All right, so we talked a little bit about ABO blood typing too. The human blood type is a genetic trait that is influenced by different alleles. It's also an example of a polymorphism because there are more than two possibilities. It's not like the monohybrid traits where you're either a tongue roller or a non-tongue roller, or you either have the ability to taste PTC or you're unable to, or you have attached earlobes or free hanging. There's only two variants for the phenotype. For ABO blood typing, there are four possible phenotypes. So you're either type A, type B, type AB, or type O. But you actually have six possible genotypes. So you inherit one of those alleles from mom and one of those alleles from dad, okay? So at that homologous chromosome, at the location, at the locus that determines ABO blood type, at each locus on each chromosome, you will inherit one from mom and one from dad. Okay, one chromosome from mom, one chromosome from dad. So the combination will determine your overall blood type. So if you have blood type A, you have two possibilities. You could have inherited a dominant A allele from both mom and dad, or you could have inherited an A from mom and an O from dad, for example. But remember that A allele is dominant over the O. So your overall blood type is still going to be blood type A, okay? So type A has two possible genotypes. You could be AA or you could be AO. Same thing for blood type B. So if you're blood type B, you could have inherited a dominant B allele from both mom and dad, or you could have inherited a dominant allele from one parent and a recessive O allele from the other. But either way, since that B is dominant over the O, your overall blood type is still going to be type B. AB, that's our co-dominant condition. So remember, co-dominant, they're dominant together. So if you inherit an A allele from one parent and a B allele from the other, they will both be expressed in your blood type, okay? You will have both A and B antigens expressed in your blood type, okay? So AB is the co-dominant condition. If you have blood type O, that means you had to inherit a recessive O allele from both mom and dad. So for AB and for O, there's only one possible genotype. The genotype and phenotype are going to be the same. But blood type A and blood type B, those are the ones that you don't know for sure. You could either be AA or AO for blood type A, or for B, you could either be BB or BO. All right, so essentially, why does all that matter? Why do we care in a class on humans, in a class on anthropology? So your blood type can be very crucial as, say, for example, you're in a situation where you need a transfusion. So if you have blood type A, you have surfaces on you have antigens on the surface of your red blood cells that will indicate the blood type. So if you have type A blood, then you have A antigens. If you have type B blood, then you have B antigens. Okay, if you have type AB blood, you have both. You have both A and B antigens. If you have type O, then you have neither one. So if you have blood type A, for example, and you receive a transfusion from somebody with blood type B, your body will immediately, there will be an immune system response, and your body will immediately recognize that B blood as foreign. And there will be what they call an antibody antigen reaction. Okay, so that's what's shown here in this image. So we call that agglutination. So when there is an anti-B, anti-A, anti-B reaction, we call that agglutination. And it's essentially your immune system recognizing the new antigens as foreign. So the antibodies will attach themselves to the foreign antigens, causing clumping of the red blood cells. And these clumped red blood cells will be unable to pass through your blood vessels. And that's a huge problem because red blood cells carry oxygen. Okay, so if those red blood cells are unable to pass comfortably through your blood vessels and reach your organs, your liver, your heart, your lungs, your stomach, your intestines, et cetera, if your brain, obviously, if that oxygenated red blood is not able to reach those organs, then that could result in organ failure and eventually death without quick medical intervention. Okay, so that's agglutination. That's what happens if you receive the wrong type of blood. Okay, so if you're blood type A, you can receive from other people that have A, or you can receive from O. You can donate to other people that have A, or you can donate to AB. Because the A's will be recognized in the AB. If you have blood type B, you can receive from other people that have B or from O, and you can donate to B or to AB. If you're blood type AB, you can receive from anyone, actually. You're the universal recipient. You can receive from A, you can receive from B, you can receive from obviously others that have AB, or you could also receive from O. You're the universal recipient, but you can only donate to others that have AB. And then if you're O, it's the opposite. If you're O, you can donate to anyone because you do not have any antigens on the surface of your red blood cells. You can donate to anyone, AB, or O, but you can only receive blood from other people that have blood type O. Okay, so you're the universal donor that you can only receive from other people that have type O. All right, questions about blood types or how to do Punnett squares with blood types. Feeling good? Okay. All right, so I've already alluded to this a bit, but the Punnett squares that we did on Monday, they are very useful for many circumstances. Punnett squares are very useful when we're talking about monohybrid Mendelian type traits, one protein, one gene type model. But a vast majority of our human genetics, not just human genetics, but the vast majority of genetics gets a little more complicated than that because most of our characteristics are polygenic, meaning that they're determined by genes at more than one locus. So remember that term locus, that's the location of a gene on the chromosome. So many of our characteristics are determined by multiple genes at multiple locations on multiple chromosomes, okay? So if that's the case, you cannot necessarily determine the likelihood of having certain genotypes of children because it's being determined by so many different factors. You would have to do multiple Punnett squares to determine the likelihood, which sometimes you can do, but sometimes it gets a little more complicated than just one Punnett square. So polygenic, so break that word up, many genic genes. So polygenic traits are those traits that are determined by genes at two or more loci, so two or more locations on the chromosomes, and also influenced by the environment. So skin pigmentation, skin color is one of those. It's determined by at least 3 separate genes and is influenced by the environment as well. So skin pigmentation is something we'll learn a lot more about. We'll learn about the evolution of it once we get to topic 5 after our first exam. But for now, know that it is influenced by multiple genes at multiple locations on chromosomes. And of course, it's also influenced by environmental circumstances. All right, so for some traits, only some of the genetic variation can be calculated. That's the inheritability. So heritability can range from 0, meaning that none of the variation is genetic, to 1, which is all of the variation is genetic. So this slide is important just because it's actually going to become more important in chapter 4 once we start learning about the forces of evolution. But for now, just know that in order for a characteristic to be influenced by the forces of evolution, it must be inheritable. It must be something that is passed down from generation to generation. If I dye my hair purple, my children aren't going to have purple hair, right? So, it's just keeping in mind that the traits that we study are those that are going to be passed down from generation to generation, because natural selection can only operate upon that type of variation, or it only influences that type of variation, I should say. All right, so the measurement of inheritability is complicated by pleiotropy, or a single allele having multiple effects. Most complex traits are both pleiotropic and polygenic. So we already talked about polygenic. So polygenic is referring to one phenotypic trait, like skin color or eye color, for example, that is affected by two or more genes. And then that physical manifestation is also sometimes influenced by the environment. So some examples here in humans: skin color, height, head form, tooth size, and overall eye shape. So those are all characteristics that are polygenic, and also sometimes their expression is influenced by the environment. So it gets very complicated, right? And then pleiotropy is kind of the opposite. It refers to one gene that affects more than one phenotypic trait. So for an example, one allele that's affecting both eye color and hair color, for example, or one allele that's affecting one gene that's affecting both height and arm length, for example. So many of what we're going to learn about, many of what we learn about in genetics, we learn about I always like to start with the simple and then make it more complex, right? So what we learned on Monday, that's kind of we learned about on Monday, that's the beginning of it. That's the beginning of understanding genetics, is understanding how these basic monohybrid characteristics operate, right? So, and it's still important. I firmly believe it is still worthwhile and important to learn about Punnett squares and learn how the basics work. But it's also important that I just reinforce and remind you that not all genetics are that simple, right? It does get a little bit more complicated. Now, we won't get much more complicated than that in here in an intro class, but I do want you to know that genetics does get more complicated than the simple monohybrid crosses we did on Monday, okay? But if you are interested, feel free to come by and see me during office hours. I'm happy to show you some more complex Punnett squares. We just don't simply have time for it here in a one semester class like this. Okay. All right. I believe that is the last slide. I believe that is the last slide for chapter 3. Yeah, it's just a picture that's going along. Yeah, we're on to our terms. Okay. So are there any questions at all? Anything about chapter 3 that anybody wants to talk about again? Or in more detail? Okay. Just for organizational purposes, I don't know what I just did there. I'm going to pause. I'm going to stop the recording and then restart it just so I can have a clear in between chapter 3.